All right, hello everyone, and thank you for tuning in for my remote defense. This isn't exactly how I imagined I'd be giving my final talk as a graduate student, but I'm really grateful for the opportunity to present my research with you all nonetheless. So today I want to share with you an example of what I think is one of the most captivating things about adaptation, when species can evolve the ability to extract new resources from their environment. I study these three rapidly diverged species of Cyprinodon pupfishes, and I want you to pay special attention to differences in their faces, because these differences tell a story about how these species are each uniquely adapted to a different ecological niche. Now, this story involves rapid speciation and ecological divergence, which are two hallmarks of adaptive radiation, which you might associate with classic examples like Darwin's finches or East African cichlids, where in both cases, a common ancestor rapidly gave rise to numerous species exhibiting divergent craniofacial morphologies adapted to different feeding strategies. For example, we know the big beaks on these finches are great for cracking large seeds, and these downturned jaws on this cichlid are great for scraping algae off of rocks. And these types of awesome matches between diet and morphology are really common across adaptive radiations worldwide and are thought to evolve due to the process of ecological speciation, which is when divergent selection favors different adaptive phenotypes and reproductive isolation evolves between populations. Now, since this process unites principles of evolution and ecology, research in these systems has been central to our understanding of mechanisms driving species divergence. However, even within the most famous and well-studied examples like Darwin's finches and African cichlids, we're only beginning to understand genetic changes that give rise to adaptive phenotypes and maintain species boundaries. To address this, my research examines adaptive radiation from a genetic perspective, asking questions using genomics and transcriptomics to examine adaptive morphology, parallel evolution, and mechanisms of reproductive isolation. And this research is motivated by longstanding questions in evolutionary biology, such as how does genetic variation translate to phenotypic variation? How predictable is evolution? And how do interactions between genetic variants contribute to reproductive isolation? So first, I'd like to begin today by describing why this study system is great for asking these types of questions. And then I'll move on to my thesis research split into three chunks. The first bit, I want to know how nature builds new traits. I want to discuss what are the types of genetic changes and changes in gene expression that affect adaptive morphology. And then I'll show you similar genetic changes between species that you might not expect looking at their morphology. And then finally, I'll discuss some evidence for a mechanism that may limit gene flow between young species by asking how selection can shape patterns of gene expression in hybrids. All right, so first I told you that adaptive radiations make these great study systems because they link principles of evolution and ecology. But why do I study this adaptive radiation in particular? Well, first off, these fishes live on some really pretty islands in the Caribbean. My lab and I would get to go down and sample in these shallow hypersaline lakes that are on the interior of these Caribbean islands, where we're looking for species like this, Cyprinodon variegatus. This species has a really wide range from the coast of the United States all the way down through the Caribbean to South America. And throughout this range, pupfishes are usually dietary generalists, like this little guy. And this means that they're mainly going around eating macroalgae, detritus, and small invertebrates. However, there are two very interesting exceptions to this rule on the small island of San Salvador, which is a tiny island only about 11 miles long, and it's covered in these shallow hypersaline lakes. Here there's been a recent adaptive radiation where we find the generalist pupfish species as well as two endemic trophic specialist pupfishes. The first of those specialists I'll talk about is the molluscivore, Cyprinodon brontothoroides. This species has evolved short, robust jaws relative to the generalist, as well as this nasal protrusion that it uses as a wedge to pry snails out of their shells. So I'll refer to this species as the molescivore, and here's a molescivore in action, where it's using its cute little nose to pop its meal out of its shell in the cutest way possible. Okay, so that's our molescivore. The other specialist species is Cyprinodon desquameter, which I'll refer to as the scale eater. This species has alternatively evolved these really long jaws, as well as this more elongated body, that allow it to remove scales from other fish in quick strike attacks. And so I'll refer to this as the scale eater. Here's the scale eater in action. This is a male scale eater going after a generalist who just gets out of the way. And notice the dark coloration of this scale eater. This is a male in his breeding territory and his breeding colors. And the breeding coloration of the scale eaters is much darker than either of the other two species. All right, so this is our cast of characters for the day, the San Salvador Island pupfish radiation. And there are several things about this system that make it very strange, that also make it a great genetic model for ecological speciation. First off, if we think back to our textbook examples of adaptive radiations, where species diverged hundreds of thousands to millions of years ago, these pupfishes diverged much more rapidly within the last 10,000 years, and yet we still see divergence in adaptive craniofacial morphology like these other systems. Another strange thing, like I mentioned, these two specialist species are endemic to San Salvador Island, and we haven't found them anywhere else, despite other islands in the Caribbean having similar lakes with similar ecologies to San Salvador. So we've searched a lot of lakes on a lot of different islands. I've searched some myself in some remote places like Mayaguana, this island, where about 300 people live. I've also searched some less remote islands like the Exumas. For example, I searched this lake here, which is behind a liquor store and across the street from where the infamous Fire Festival was supposed to take place. But my point here is that even this lake looked similar to lakes that you'd see on San Salvador. And yet, I only found generalist pupfishes. And this is true of every other lake on every other island that we've searched so far. So what's really special about San Salvador is we find all of these hypersaline lakes, and many of these lakes contain all three species living amongst one another in sympatry. So this is exciting because it suggests that there are reproductive barriers between species, maintaining these species boundaries. And they've evolved significant pre-mating isolation. What this means is if we were snorkeling around these lakes and looking down from above, we would often see mating like this, where a male courts a female of his own species, and they do their little mating wiggle. However, we do sometimes see interspecific mating between species, and when we bring all three species back to the lab, we can cross them in all directions and they produce fertile hybrid offspring. When we do cross these species, they tend to show intermediate craniofacial phenotypes, which means that most F1 hybrids end up looking kind of like generalist species. All right, so what makes this system great as a genetic model is that we have rapid speciation between these species that are living in sympatry and are still able to hybridize, and yet despite their young age, these species show drastic divergence in craniofacial morphology and other adaptive traits. And so for the rest of this talk, I'll refer to the generalist. I'll show the generalist in red, I'll show the molescivore in green, and the scale eater in blue. All right, so we have this dramatic craniofacial divergence between species, leading me to my first question of what are the types of genetic changes required to build these novel traits in the specialists? And so to get at this question, I had 58 whole genomes. Most of those genomes were from individuals from San Salvador Island. I also had some generalist outgroup populations from throughout the Caribbean, as well as a couple of outgroups from Mexico and Death Valley in California. I used a standard variant calling pipeline to identify over 13.8 million SNPs across these individuals. And like I said, most of these individuals came from San Salvador Island. So one of my first questions was, out of these millions of SNPs across individuals, are any of them unique to either of the specialist species? And so to look at this question, I measured FST, which is a relative measure of genetic differentiation that ranges between 0 and 1, where 1 indicates that an allele or a SNP is unique to a population, also termed fixed between populations. So looking at these different pairwise comparisons, the specialists are the most diverse from one another and showed the highest number of SNPs fixed between populations, over 6,000 SNPs. There were fewer fixed between generalists and scale eaters, only 1,500, and just 79 fixed between generalists and molluscivores, which is a very low number of SNPs fixed between species, showing this dramatic divergence in phenotype. And to put this into perspective, we can look to bird genomes that have been sequenced, where between golden and blue-winged warblers, there's about 74 SNPs fixed, and between hooded and carrion crows, there are about 82. And my main point here is that the primary difference between these species pairs is plumage color or pigmentation differences. My results are suggesting that a similar low number of SNPs are fixed between species showing drastic divergence and complex traits in these craniofacial phenotypes. And so if my question was, can we identify genetic variation associated with differences in these adaptive phenotypes, we figured it might be a good place to start looking at regions of the genome containing these SNPs that are fixed between populations. So this is an example of two genomic regions containing SNPs fixed between the two specialist species. This region doesn't have any genes annotated. This region has a gene called GALR2. So next, I looked across each species genome looking for evidence of a hard selective sweep, where selection favored a single haplotype rising quickly to high frequency in a population. So we can imagine a hypothetical ancestral population of chromosomes represented by these lines, and each of these empty circles representing an allele or a SNP that's neutral. If one day we had a beneficial allele pop up on this genetic background, over time selection and recombination might shape a population of chromosomes that look like this, where our beneficial allele affecting some adaptive trait has become fixed in the population, dragging with it this neutral variation and shifting the site frequency spectrum towards high and low frequency derived variants. So looking for these patterns across each species genome, I found that these regions containing SNPs fixed between the specialists also showed evidence for a hard selective sweep in the scale eater populations. I also looked at patterns of within population nucleotide diversity, as well as absolute genetic divergence between populations, to verify that these regions showed signatures consistent with selection and not confounding processes like low recombination rates. And then finally, I performed genome-wide association mapping, asking whether I could find regions of the genome that seemed to be associated with variation in jaw size between species. So I used two approaches, one Bayesian and one Frequentist, and found that there were a lot of regions of the genome implicated as significantly associated with jaw size by both strategies. This is what the output of the Bayesian approach looks like, where these gray and black alternating correspond to the 9,000 scaffolds making up our reference genome. And these points here represent 20 kb windows showing the strongest association with variation in jaw size between species. So overlapping the candidate regions that were associated with jaw size with regions showing signatures of selection, I found a total of 31 genomic regions, so 20 kb regions, that contained 18 candidate genes that had never previously been associated with craniofacial phenotypes, and 11 genes that were annotated for skeletal system effects from other model systems. And so this was really exciting. One really exciting thing about this region in particular, containing the gene GALR2, is that it falls within a previously identified QTL, explaining up to 15% of the variation in jaw size between species. And another interesting thing about this region is that out of all these SNPs that are fixed here at a FST of 1, none of them are falling within the coding region of the gene. And this was true of a lot of the candidates that I identified. And so that suggested that the next piece of this puzzle was to ask, are these variants fixed between species influencing differences in craniofacial morphology or other adaptive traits through changes in gene expression? And so to get at this question, we'll go back to San Salvador, where I again sampled individuals of all 3 species. This time I sampled total mRNA. So I set up breeding tanks with individuals of the same species from the same lake population from San Salvador, collected their eggs and let them develop for 8 days. And at this point, I extracted whole body total mRNA for sequencing. And then we also let some individuals grow up to 20 days old. And at this stage, they're large enough that I can take some forceps and dissect craniofacial tissues and specifically extract RNA from those tissues. So then I used a standard gene expression R package, DESeq2, to identify genes that were differentially expressed between the generalist species and each of the specialist species for both of those developmental stages. And I found a lot of genes here in red that were significantly differentially expressed between the generalist and each of the species at both time points, with the most expression divergence at this 8-day-old stage between generalists and scale eaters. Now, for the exciting part was overlapping those genes that I identified that were associated with jaw size variation and showed signs of selection. I found that a lot of them, 11, were differentially expressed in at least one of these comparisons, including these genes in bold that are annotated for effects on skeletal system development. And so this was very exciting, and I'd like to come back to this data because there's a lot to pick apart here. But first, I want to summarize what this information tells us about the genetic basis of adaptive morphology in this system. So first off, we found that there were very few variants fixed between species, and that some of those variants were associated with divergence in these adaptive craniofacial traits and near genes known to affect skeletal development. And then the gene expression data showed us that many of the candidate genes identified in this approach were differentially expressed between species, suggesting that maybe this fixed variation is influencing differences in gene expression that ultimately influence differences in craniofacial morphology. All right, so now I'd like to come back to this data set because there's an interesting and unintuitive pattern here that only reveals itself when you look at the intersection of comparisons. I want to talk about parallel evolution of gene expression in this system. So we know from many examples in nature that when species are faced with similar selective pressures, they often respond with the same adaptive solutions. Normally when we think about parallel evolution, we think about things like color or morphology, but we can also think about parallel evolution at the level of gene expression or at the level of nucleotide changes. And studying these types of systems can teach us about evolutionary constraints and also potentially help us to predict evolutionary trajectories. Now, there's been a lot of research showing that parallel genetic changes tend to control parallel niche specialization, but there's been less research about parallel genetic changes underlying divergent trophic specialization. So that's why I'm interested in coming back to this data, is I want to look at the potential for parallel evolution in this system. So if we take this 8-day-old information and simplify it a bit, I found about 6,000 genes differentially expressed between the generalist and the scale eater, and only about 1,000 differentially expressed between the generalist and the molluscivore. Now, looking at the overlap between these comparisons, there seem to be a lot of genes that are uniquely differentially expressed between the generalist and scale eater, but then just this little sliver that are uniquely differentially expressed between the generalist and the molluscivore. And then in this overlap region in black, these are genes that show differential expression in both specialists relative to the generalist. And this was an immediately surprising amount of overlap. In fact, 80% of genes differentially expressed between generalist and molluscivore were also differentially expressed between generalists and scale eaters. Now we wanted to look at the direction that these genes were being differentially expressed in both specialists because it could tell us something about selection acting on gene expression. So if we were to grab a gene randomly from this overlap, it could show a couple of different directions of expression. Either it is showing the same direction of differential expression, where maybe it's upregulated in both specialists relative to the generalist, or downregulated in the same direction, or it could show opposite directions of differential expression, being upregulated in one specialist relative to the generalist and downregulated in the other. Now, a neutral model of gene expression evolution might suggest that out of these roughly 800 genes in this overlap, we'd expect to find about 400 showing each direction of differential expression. But what we actually find is this. Almost every single gene that's differentially expressed in both specialists relative to the generalist is differentially expressed in the same direction. Meaning if I were to randomly grab a gene out of this overlap and it was upregulated in scale eaters, chances are very likely that it's also upregulated in the molluscivore. And this is exciting because it's the first case of a very strange pattern. We see significant parallel evolution of gene expression between divergent trophic specialists. Now, why is this weird? This is weird because I've told you about all the ways that these species are divergent from one another in their craniofacial morphology, in their different feeding behaviors, in their male breeding coloration. And so this really begs the question, what do these divergent specialist species have in common that might arise from similar changes at the level of gene expression? So one potentially straightforward answer is that the specialists are more closely related to each other than either is to the generalist species. But if we look at a phylogenetic tree of all of our genomic samples, it looks like the scale eaters form one group, whereas the generalists and molluscivores tend to group by lake population from these different lakes on San Salvador. And so this suggests that parallel evolution of expression is not due to a more recent split between the specialists. Another potential answer might come from gene ontology enrichment analyses. So looking at these genes in the overlap of those two comparisons, the genes showing parallel expression in specialists, these genes were highly enriched for metabolic processes. Looking at the genes that showed divergent expression patterns that were unique to each specialist, these genes were also enriched for metabolic processes, but they were also enriched for these other categories, including cranial development and pigment biosynthesis, these phenotypes that are more obviously different between the specialists. And so this might begin to paint a picture where parallel changes in gene expression may be parallel adaptations to higher trophic levels, where scale-eating and molluscivore niches present similar metabolic requirements and a more nitrogen-rich diet relative to the lower trophic level of the generalists. So this plot here was from a previous study that used stable isotopes from dissected muscle tissues to identify relative trophic position across all three species. And we can see that the scale eaters occupy the highest trophic position with the highest nitrogen-rich diet, followed by the molluscivore, and then finally the generalists occupying the lowest trophic position. And so I hope that the main takeaway here is that we're showing what is, to our knowledge, the first evidence of parallel changes in gene expression underlying divergent trophic specialization. But then upon closer inspection, these parallel changes seem like parallel adaptations to higher trophic levels presented by these novel niches. And then this last point here, we see that the unique patterns of expression divergence that were unique to each of the specialists seem more important for these differences in more obvious phenotypic differences like craniofacial divergence and pigmentation differences. And it's these types of genes I want to talk about for the last part of my talk, genes influencing divergent adaptive phenotypes. Only I want to think about them in the context of hybrids and how they may contribute to reducing gene flow between species. So, ecological speciation theory suggests that divergent ecological selection can lead to the evolution of new adaptive traits, and that reproductive isolation can evolve between those populations as a byproduct of adaptation. And we can also think about this at the level of gene regulation, where cis and trans regulatory factors bind to control the expression level of some gene. If we have two populations that are adapting to divergent niches, over time, divergent selection may favor cis and trans regulatory factors that cause low expression of a gene in one species and high expression of the same gene in another species. Now, if these species come back into contact and form F1 hybrids, these regulatory alleles will become decoupled because they were co-adapted in each species. And this decoupling of co-adapted regulatory factors could lead to expression levels that are much lower or much higher in the F1 hybrids compared to either parental population. This expression pattern is known as hybrid gene misregulation, and it can be caused by these types of Dobzhansky-Muller incompatibilities between divergent regulatory alleles. So now I can kind of refine the question that I'm asking for this section of my talk is, can divergent ecological selection drive hybrid gene misregulation? If it can, then we should be able to identify genes that influence divergent adaptive phenotypes that are differentially expressed between species and are also misregulated in their F1 hybrids. So this isn't a new question, but it remains unresolved. A lot of what we know about hybrid gene misregulation comes from studies on distantly related species and subspecies of Drosophila and mouse, where in these systems, gene misregulation is associated with strong intrinsic reproductive isolating barriers like hybrid sterility and inviability. However, it's debated whether misregulation is the cause or consequence of these strong reproductive barriers. Furthermore, gene misregulation in these systems is thought to evolve due to stabilizing selection in allopatry, not divergent selection in sympatry. And that's why I want to look at hybrid gene misregulation in this pupfish system. So to look at this, I generated 124 new transcriptomes. So forget about all of the gene expression data I was talking about before. These are new transcriptomes, and they're derived from developing samples from San Salvador Island, from those sympatric populations of generalist molluscivores and scale eaters, as well as outgroup generalist populations. So one population from New Providence Island, which is about 300 kilometers away from San Salvador, and one generalist population from North Carolina, which is about 1,000 kilometers away from San Salvador. So first I wanted to look for patterns that I already knew existed from the other gene expression data, looking for patterns of differential expression between species. So I again set up breeding tanks containing individuals from the same lake populations or the same generalist outgroup populations of the same species, collected embryos and let them develop again for 8 days. So looked at the same whole body tissue at 8 days. But I also looked at this earlier developmental period at 2 days. And at this stage, we know that cranial neural crest cells are migrating to the pharyngeal arches, which is where a lot of craniofacial magic happens early in development. So I have these two developmental stages that are early developmental stages. And I again generated read counts and asked again, can I find genes that are differentially expressed between populations? So for example, I found over 4,000 genes differentially expressed between the molluscivores and the scale eaters, the specialist populations, at the 8-day-old stage. And when we look at the total number of genes expressed, that's a lot of genes differentially expressed. That's about 30% of all genes expressed or differentially expressed between the specialists. Now on the x-axis here, I'm looking at phylogenetic distance between populations using branch lengths from that tree I just showed. And this is because I want to understand the relationship between gene expression divergence and genetic divergence between populations. And this is because we have some nice predictions about how that should look. So we know that between closely related populations, gene expression divergence tends to increase with phylogenetic distance under genetic drift until it reaches a boundary where stabilizing selection constrains gene expression divergence. So relating that to predictions for this system, we have these allopatric generalist populations, and these populations are all adapted to similar ecological niches. And we might expect that genetic drift is a strong force acting on gene expression divergence between these generalist populations. Alternatively, if strong selection on gene expression has been important during adaptation in these divergent niche specialists on San Salvador Island, we might expect to see a more rapid rate of gene expression divergence between these sympatric populations on San Salvador Island. So looking at some of the other comparisons here, I found a low amount of genes differentially expressed between the San Salvador Island generalist and the molluscivore, as well as the San Salvador Island generalist and the scale eater. And then comparing the San Salvador Island generalist to those two outgroup generalist populations, I found that there were about 10% of genes differentially expressed. But what I hope is the main outlier here that you can see is this specialist comparison. There's more genes differentially expressed between these specialists that are living sympatrically compared to even generalist populations that are separated by over 1,000 kilometers. And so this is a really strong indicator that selection on gene expression has been important during adaptation to these divergent niches in the molluscivores and the scale eaters. All right, so this is consistent with my first prediction here that we should find genes differentially expressed. Now let's look for patterns of hybrid gene misregulation in the F1s. So to generate these F1 hybrid transcriptomes, I again set up breeding tanks, but this time between species, and then collected eggs and extracted RNA at the same developmental stages that I discussed previously. But I also, in this case, extracted and sequenced the genomes of the parents that were used to generate those F1 hybrids so that I could determine cis and trans regulatory mechanisms underlying expression divergence between those parental populations. And so my goal here was to take these F1 hybrid transcriptomes and compare them to the purebred F1 transcriptomes from their parental populations and ask, can I find genes that are differentially expressed? And there was some question as to whether we'd find any gene misregulation in this system, but in fact we did. So there were about 1,500 genes differentially expressed between the F1 hybrid transcriptomes generated from molluscivores and scale eaters. And so if we again look at the total number of genes misregulated here, that's about 10% of genes misregulated in these specialist hybrids. Now, I again wanted to ask whether this pattern of gene misregulation was a result of selection on gene expression. And so I wanted to think back to the predictions that we had for gene expression divergence and think maybe we should expect something similar for hybrid gene misregulation, where as you increase phylogenetic distance between parental populations, you'd expect more hybrid gene misregulation. So looking at some of those other comparisons, I found that the hybrids generated from crosses between San Salvador Island generalists and molluscivores showed very low misregulation, where the cross between San Salvador Island generalists and scale eaters was pretty high to that compared to that specialist comparison, around 10% of genes misregulated. Now looking at those outgroup generalist hybrids, I found very low misregulation in the New Providence Island cross, but very high misregulation in the North Carolina cross. And this was a little confusing to interpret because if you were to cover this point, it looks like hybrid gene misregulation is evolving due to our expectations under genetic drift, with increasing misregulation with increasing phylogenetic distance. But if we cover this point, it looks like these scale eater cross hybrids, that misregulation is evolving more rapidly than we would expect under drift. And so I wanted to think about this data in a slightly different way. So now this is the exact same data, but the x-axis is now kilometers of distance between populations. And I used branch lengths from that tree to perform a phylogenetic least squares regression and found that hybrid gene misregulation did not seem to be explained by geographic distance between populations. So what this indicates to us is that these high rates of misregulation that we see in the scale eater crosses is higher than we would expect after accounting for phylogenetic relatedness across samples, suggesting potentially that selection on gene regulation has driven this pattern. So now we have evidence for selection acting on gene regulation using this phylogenetic framework. But another important prediction here of my question, can divergent ecological selection drive hybrid gene misregulation? Another important prediction is that the same gene that's differentially expressed between species should also show misregulation in the F1 hybrids. And so, excitingly, I found that there were many genes that were differentially expressed between species and also misregulated in their F1s. For example, these genes in purple here are differentially expressed between molluscivores and scale eaters and also misregulated in their F1 hybrids. And I found more than just these 320 genes in this overlap here when I looked at crosses between the generalist and each of the specialists. So that in total there were 750 genes matching this expression pattern. So next, I wanted to know if these genes showed signs of selection. So as a first pass, I looked to see if these genes were near SNPs fixed between populations, as well as within regions showing high between population divergence measured as DXY. And I found that 125 genes matched this expression pattern, or match these criteria. And I'm referring to these 125 genes as ecological DMI candidate genes. DMI because they show expression patterns consistent with divergent regulatory alleles negatively interacting to produce misregulation in the hybrid, and ecological because these genes were strikingly enriched for developmental processes related to trophic specialization. For example, we found enrichment related to craniofacial development, related to skeletal muscle development. Here I'm showing that specialists tend to have larger adductor mandibulae muscles, which are muscles important for the force of jaw closure. We also saw enrichment related to nitrogen metabolism. As I explained earlier, we know that the specialists occupy a higher trophic position with more nitrogen-rich diets. And then we also saw enrichment related to brain development, which could affect a number of different behavior differences between species. One of my favorite examples that my lab mate Michelle has shown is that the specialists tend to be more aggressive than the generalists. Okay, so we have all of this enrichment information showing that these genes seem to have some adaptive potential, but we can go beyond gene ontology enrichment to show that these genes may be involved in adaptive processes. For example, this gene MPP1 is significantly differentially expressed between the generalist and the scale eater species, and also misregulated in their F1 hybrids, which show lower expression. This gene has recently been shown, mutations in this gene have been shown to influence craniofacial deformities in mice and humans, and knockouts of this gene in mice show decreased bone density. This gene also shows expression patterns consistent with cis regulatory divergence. So in the F1 hybrids, we always see that the scale eater allele is more highly expressed than the generalist allele, which in short is really cool because it suggests that there's some linked nearby variation to MPP1 that's controlling expression differences between species. And we find 170 SNPs that are fixed between generalists and scale eaters within 20 kilobases of the MPP1 gene region, shown in blue here, and each one of these dots corresponding to a SNP that's fixed between species. This region also shows signs consistent with selection with high between population divergence and low within population diversity. It shows that characteristic shift in the site frequency spectrum associated with a hard selective sweep in the scale eaters. And it's also significantly associated with jaw size using that genome-wide association mapping approach I described earlier. And so together, this suggests that MPP1 and other candidate genes like it are differentially expressed between species due to their influence on the development of adaptive traits. What then does this mean for the hybrids where these seemingly important genes are misregulated? Well, first I want to back up and point out a couple of really cool things we know about this system. First off, we know that hybrids that show craniofacial phenotypes that are intermediate between parental types show low fitness in the wild, suggesting that extrinsic ecological factors are an important source of reproductive isolation in this system. We also know that from recent feeding kinematic data that F1 hybrids differ significantly from their scale-eating parents in traits that are essential for scale eating, like peak gape of the jaws, as well as gape angle of the jaws. So, putting all of this together, if we have divergent selection acting on a gene so that it shows divergent expression between species, and that same gene shows misregulation in F1 hybrids, then perhaps that misregulation causes weak intrinsic isolation in the F1. Alternatively, or additionally, it's not mutually exclusive, this process could lead to, the misregulation of this important gene could lead to abnormal development of craniofacial phenotypes that result in suboptimal feeding kinematics in the F1 that ultimately cause reduced fitness in the wild, causing extrinsic isolation. Now I have one more piece of information that supports this middle link here, and it comes from expression data from 20-day-old hybrids from crosses between generalists and molluscivores. Now remember, at this 20-day-old stage, the fish are large enough that I can dissect craniofacial tissues, and what I found is that these craniofacial tissues showed really high rates of misregulation, much more than we see at the 8-day-old stage. And so my takeaways here are that I've shown you misregulated genes under selection seem to be associated with adaptive ecological traits in these trophic specialists. But I'm hoping that maybe the broader takeaway here that you're thinking about later is that these evidence support a compelling genetic mechanism of rapid speciation, where misregulation can be driven by these adaptive processes and may contribute to reduced hybrid fitness, ultimately contributing to reproductive isolation between young species. And with that, that's all my thesis research. And this is the part where I turn into a full-on YouTuber and say, if you'd like to hear more about these projects I discussed today, I've got links to my manuscripts underneath. And I'm also going to try to take questions in the comments box. So I'm going to go into acknowledgements now, but if you have a question that's fresh in your mind, go ahead and type it in, and then I'll thank my funding sources and the people that got me here in the meantime.